So here roll up another tumor suppressor gene that is also called the guardian of the genome, because it play a major role in repair mechanism and also apoptosis. So here the tumor suppressor gene or protein name is p53. The protein is p53. So here the function, function of p53 in cell cycle control. So here is a function of p53, how p53 works in control of cell cycle, and what are the proteins that are involved in this process. So first, in case of normal cell, so first we discuss on normal cell. So in case of normal cell, so in case of normal cell, so first we discuss on normal cell. So in case of normal cells, all the functions which occur are remain in a regulating manner, or cell cycles occur in a regulating manner. Several proteins are involved, and those proteins are regulated in a those proteins are works in a regulations manner, and ultimately cell divide and proliferations of the cells occur. But a programmed cell divisions occur, actually the programmed cell divisions. So in case of when there is no DNA damage or no any mutations in the DNA that needs to repair, in this case p53 binds with another protein. The name of the protein is MDM2. So MDM2. So MDM2 is a protein that play a major role in inhibitions of functions of p53. And here the p53, so it inhibits the functions of p53. So p53 is a tumor suppressor. So here is p53, so it is p53 protein. So when MDM2 binds with p53, so when MDM2 binds with p53, it stimulates degradations of p53. So role of MDM2 is to inhibit the functions of p53 by its degradations. So when p53 it binds with MDM2, it leads to its degradations. So what is the role of the MDM2? So MDM2 stimulates degradation of p53. So when the p53 when the p53 is degraded by MDM2, the amount of p53 becomes low. in case of normal cell, because there is no need of p53. P53 is a transcription factors and helps in the activations of several genes, helps in the activations of some repair genes that repair ultimately repairs protein form, and ultimately repair of the DNA is occurred. But when there is no DNA damage, no such type of mutations in the DNA that needs to repair, in this case p53 is degraded by MDM2. That is why the amount of p53 is low in case of normal cell. But now cell with DNA damage. Now, so this is case 1. So this is case 1. Now case 2. In case of 2, that is in cell with DNA damage, cell with DNA damage. So now cell with DNA damage. So in this case, so what happened? So induction, suppose this is DNA, you can even imagine it is DNA, because this is not a structure of the DNA, you have to imagine it. So induction of DNA damage. So first there is inductions of the DNA damage, means DNA damage occur. Suppose particular this position, at particular these positions DNA damage occur or any mutations occur, and that lead to phosphorylations of both protein, both MDM2 and p53 protein. So DNA damage leads to phosphorylations. So DNA damage leads to phosphorylation of both p53 and MDM2 and p53. So when you draw the pictures of anything, it remember faster in your mind and it remain for a long time. So try to draw the mechanism of actions of any protein, that it can remains in your mind in a long term, and draw it in your itself, because you have to draw yourself. Don't try to copy, don't try to just read it. You have to just draw what is the functions of the proteins. Diagram, you have to draw the diagram by any type of chart. Make your own chart, so it can remain in your mind for a long time and you can easily understand, though it takes long time, but it is helpful for you. So thank you. Now MDM2, so phosphorylated form of MDM2, phosphorylated form. So this is MDM2. So phosphorylated form of MDM2, this is MDM2, and this is p53. So this is p53, another is MDM2. So phosphorylated form, they cannot, so phosphorylated form of MDM2 combined with the p53, and now p53 is free. So p53 is a transcription factor. So what is the role of the transcription factor? The role of the transcription factor is to stimulate expressions of the some genes. So now p53, this is called activated form of the p53, or now p53 attached with a DNA and helps in the expressions of a particular gene, that is called WAF1. So phosphorylated form phosphorylated form of p53 act as a transcription factor and helps in the expressions of WAF1. WAF1 encode a protein. WAF1 encode a protein that is called p21 protein. So what is the name? p21. 21 is the molecular weight of that protein. So p21 is a protein. p21 is a protein that is encoded by WAF1 with the help of a transcription factor p53. Now what is the role of the p21? p21 blocks the functions of a complex known as Cyclin D CDK2. So Cyclin D and CDK2. So this is Cyclin D and this is CDK2. So p21 blocks the function of Cyclin D CDK2 complex by binding with them. So this is p21. Now p21 binds with that complex and inhibit the p21 Cyclin D and CDK4 complex. So what is the role of the Cyclin D CDK complex? Cyclin D and Cyclin D CDK4 and Cyclin E CDK2 helps in the progressions of the phage from G1 to S. So Cyclin D and CDK4 helps in the phosphorylations of PRB. Now if phosphorylated form of PRB does not bind with E2F, and E2F is also a transcription factor, helps in the progressions of a cell from G1 to S phase and ultimately initiation of replication occur. But when we inhibits the functions of Cyclin D CDK4, it cannot phosphorylate PRB. So now PRB inhibit the function of E2F, so the cell arrest in G1. So when we inhibit the functions of p53, there is no kinase activity. So no kinase activity of Cyclin D and CDK4. There is no kinase activity. So absence of kinase cause PRB not phosphorylated and E2F is therefore not activated. So now PRB is not phosphorylated, and it inhibits the function of p53, functions of E2F. So it inhibits the functions of E2F and ultimately cell arrest in G1. So ultimately cell arrest in G1. So ultimately cell remain arrest in G1 until the repair process is completely done. So this is the process by this is the role of the p53. p53 also helps in the apoptosis of the cells if the DNA damage is is not repaired by the repaired mechanism. Now here chart that through which you can easily understand. So what are the role of the p53 and so this is the chart. Like hyperproliferation signal, what is the role of the hyperproliferation signal? If the DNA gene amplification leads to hyperproliferation signal, so hyperproliferation signal DNA damage telomere shortening, we discuss about the what is telomere shortening. And hypoxia is a conditions when your body or any cell does not get enough oxygen. So oxygen deprived conditions is known as hypoxia. Detail of it we discuss later. So these 4 conditions helps in the activations of p53. So now p53, activated form of the p53, helps in cell cycle arrest if the DNA cannot repaired by the repair mechanism. So it leads to apoptosis, or ultimately it undergo a senescence. Irreversible. What is senescence? Irreversible cell arrest. Irreversible cell arrest. So when you can't repair, so when the p53 can't repair the DNA damage, so it leads to a permanent arrest of the cell cycle, and this is called as senescence. So this is the whole the process, how activations occur, which type of factors helps in the activations of p53, and what is the role of the p53, and in what mechanism p53 occur. Apoptosis is a important topic we discuss in the cell death chapter. So when we discuss about the cell death, we discuss about apoptosis because there are several proteins are involved. So this is the whole process. If you like the video, please share. Thank you.